Okay, up next we have Dr. Claire McWhite, another early career speaker for today's symposium. And Claire recently finished her PhD at the University of Texas and has now moved to Princeton University, where she's a Lewis Singler Scholar at the Institute for Integrative Genomics. And today she's going to be talking about mapping conserved protein complexes across plant species. With that, I'll let you take it away.
Okay, so today I'm going to talk about my major PhD work, which was all done in the lab of Edward Marcotte at the University of Texas at Austin. So mapping conserved protein complexes across plant species. So first off, I'm going to define what kind of protein interaction I'm talking about. This is specifically the protein complexes, which is a stable set of proteins that have some type of distinct geometry and form a functional unit. So in this case, I'm not looking for like transient, like kinase type interactions or similar. This is only two proteins that are stably bound together pretty much all the time. So many plant proteins are uncharacterized, and protein interactions a kind of guilt by association way of getting at protein characterization. So if you have a protein that you know what it's doing and you find it interacting with a second unknown protein, you have a really good hypothesis for what this second unknown protein is doing. So protein complexes tend to be conserved across multiple organisms. They don't evolve particularly quickly or change particularly quickly from what we can tell. So that means that we can use multiple species to create a consensus set of the protein complexes that are found across plants. And so in my experiment, we surveyed across all of these 13 species to try and get at biochemical signals of these common, deeply conserved biological assemblies, such as the proteasome or the CCT chaperoning complex, things like that. Okay, so the technique I'm using is protein complex discovery by cofractionation mass spectrometry, or protein correlational profiling, which you've already heard a few talks kind of about today. So the idea is that in a cell lysate where we've disrupted the membranes and it's kind of floating around soluble proteins, proteins that are in a complex together will travel through a column and drip out of the column at the same point across this 96 well plate. And then we can take each well and do mass spectrometry to determine what proteins are in each well. So the advantages of this type of technique, especially for plants, is that you don't need any type of affinity tagging, no antibodies. You can apply it to any sample with little asterisks on that. There are some samples like coral where it seems to not work that great, and pretty much any organism. So this is the full survey of the data set. We have 13 species. We've done 35 separations, which is each one of those like 96 well plates. And that makes 2,100 fractions that each have full protein identification. We selected, oh,
Sorry,
hello?
you're on the second slide still? I'm not sure if that's the intention.
Oh no, it's not. Okay. Slideshow. Is it changing now?
No.
Now we're on the slide 4.
You might want to stop sharing and reshare.
Yeah. Sorry about that. I thought I could use Zoom by now. What about now?
It's there we are. We're on slide 3.
Sorry about that. Okay, so I already talked about the gateway to functional characterization. So this is our full experimental set of species. Okay, and we selected a range of tissues to try and get into the type of protein complexes that you're going to find across any cell, any species, no matter where you look. And altogether, this was a set of about 14 million peptide mass spectra. So the fun thing about plants compared to the application of this technique to like humans or yeast types of cells is that the protein identification from peptides problem just becomes a huge issue. And this is due to many plant genomes are highly complicated and have an extensive history of whole genome duplication. So if we look at kind of a really simplified overlook of identifying proteins from peptides, consider a set where you've got protein A and protein B. They both have completely different sequences. You've adjusted them into peptides. And when you identify the peptides, you can pretty clearly tell that protein A is present in the sample and protein B is present in the sample. But it becomes complicated in organisms like wheat, which is highly duplicated, has around 140,000 protein coding genes, and is hexaploid. And so you might have a case where you've got 5 very similar, very few distinguishable peptides. And then when you detect a few that are common to most of them or all of them, it becomes a really difficult inference problem that standard approaches kind of totally fail at this task. And in practice, kind of randomly assigned to different isoforms or paralogs of this particular protein A. And so what I decided to do was to incorporate evolutionary information into our protein identification. So we know that all of these paralogs, these similar proteins in wheat, descend from some ancestral protein A. And so we can then identify that as an ortho group. And when we detect this common peptides, we say we identify ortho group A, not a particular protein within that ortho group. This has a lot of advantages to doing a multi-species comparative plant analysis. The first off was that the fold increase of spectral counts, particularly of the higher ploidy species like wheat and soy and quinoa, were substantially increased. We had way many more usable, uniquely assignable peptides. Another thing is mapping to these ortho groups provides a common key for data integration across these species that have wildly different numbers of genes. Okay, so now another overview, since you missed my first slide, of how this technique works. So we've got our plant. We have ground it up into lysate in such a way that breaks up the membranes, but preserves the intact protein complexes, and then separated that lysate across some gradient. And so the information that we can see from the mass spectrometry is its vector of detection of where each protein in that fractionation was detected. And so finding proteins with similar peptide is a point of evidence that there is some existence of a complex ABC. So I'm going to show you some real data of how actual protein complexes going across fractionation columns look like. So this was from a size exclusion of Arabidopsis sprout. And you can see that these 3 members of the proteasome have highly correlated profiles. And the CCT chaperone also has internally consistent profiles. If we do an ion exchange chromatography, which is a different type of separation, you once again see internally consistent fractionation. And we do a different type of tissue, this time seed, you once again get internally consistent profiles that are matched within protein complexes. And it's this type of orthogonal technique that is really powerful. A lot of things are about the same size and a lot of things are about the same charge, but way fewer things are both the same size and charge, and the same size and charge across multiple different species. We did cross-linking fractionation on all of the fractions for some select experiments to show that where we're seeing this correlation of a peak is the same point where we're seeing intra-complex reasonable cross-links. So we did that on soy and also Chlamydomonas. And so here for this portion of photosystem 2, we're seeing the internal cross-links in the solvent exposed area of photosystem 2 in the same fractions. Okay, so these are the 3 fractionation experiments I showed you from Arabidopsis. Because we have this common key of this orthogroup identifier, or all the paralogs of PSB1 across all the species, we can then concatenate onto all the species into one long profile, which is 2100 fractions. And all of these vectors are kind of a signature of what it looks like to be a proteasome or a member of the CCT chaperone in our experiments. So we already know that proteasome and CCT chaperone exist, but the whole point of this is to find new complexes, other sets of internally consistent correlation profiles. So we've got our vector of detection across all these 2100 fractions, covering 2400 orthogroups. And so if we zoom into some other known complexes, like the IF2B, the prefoldin, you can again see this internal consistency of subunits, and also the power you get from doing 2 different types of separations to distinguish things. So then we go into machine learning, where we're pretty much scoring how similar 2 protein solution profiles are. And this is all based off of gold standard known interactions, which you can use. It trains the model that says, okay, these 2 things are so similar, they're behaving like a stable interaction. With that, we can give each pair of proteins a score, and using a fully withheld set of known complexes that the model never saw, calculate false discovery rates. And so we can say at our cofractionation score of above 0.5 or more, that's about 90% true positive protein interactions. And depending on your application, you might be willing to have like a little lower rate, like 50% of the things are true interactions. And so this is you can define which threshold you feel like using. Okay, so taking all of these interactions, I could then hierarchically cluster those into protein complexes. And so this is a hierarchical view of protein complex assembly. You can see, for example, one level of clustering captures the 60S, another captures the 40S, and a larger captures the entire ribosome. This plot is colored green, are known annotated complex members in plants. If it's yellow, it is a novel complex members. And so we have multiple complexes that are entirely composed of proteins that are not known to be in complex with each other. The things in darker circles are uncharacterized proteins in plants, which means they come with no annotations in Uniprot or Arabidopsis on their function. Some of them you can actually figure out what they are based on homology, but we used a pretty loose definition. And so you can see a lot of highlights of the protein complex world, like COG, the ribosome, CCT, etc. But there's a lot of new stuff in here. So to end up, I'm going to show 2 examples of validating these interactions by matching phenotypes. And this is based on the principle that you have 2 proteins, you know each of those proteins causes some phenotype. When you find those 2 proteins interacting, you can then say that the malfunction of that protein complex AB is what's leading to the phenotype. And this is how a lot of kind of protein complexes were classically discovered in mutant screens. You had 2 different mutants that both created the same phenotype. It was then figured out that those 2 different phenotype causing mutations were in complex proteins. So the first one is this new complex of this protein law and this protein called domino. And they're both known to have nucleolar defects. And we confirmed that in Arabidopsis mutants. So here we're looking at the top down of Arabidopsis salique seed pod with the seeds on either side. And domino, which is why it's called domino, causes this type of alternating white-green phenotype. And law also causes that phenotype. And so what we can say is it's the complex of law and domino whose malfunction is leading to this embryonic lethality. Because law is a conserved eukaryotic protein and pretty well known, we know that it binds to the polyutils of RNA polymerase 3. We suspect that domino is also involved in the same process. And this is a really exciting complex for me because law is conserved across eukaryotes and domino is a plant-specific protein. And so this is some complex that formed in plants after eukaryotes. And so that's really interesting. So another one was predicting a novel phenotype just from protein interaction. And so this is a interaction of two mitochondrial proteins, VDAC2 and 3-beta-HSDD. This is a view of a wild type Arabidopsis main stem. And so you can see these ciliques extending off of the stem, and this is what they look like zoomed in. VDAC2 knockout is known to cause the fertility defects where you get dwarfed plants, and short ciliques. And our knockout of 3-beta-HSDD also nicely caused this same fertility defect where you can see a highly affected plant and a less affected plant that is able to kind of recover later. So this was a direct, large-scale experimental determination of the set of conserved protein complexes of plants. I think I came up with it's a new framework for dealing with proteomics of high ploidy species and incorporating this outside evolutionary information to simplify your protein identification problem. And so what's nice is after you've simplified, you can then look within that group for the specific peptide evidence for the members of that ortho group. And there's also a lot of new biological systems to investigate that we've barely started to dig into. So for resources, we have the main paper at the top. We just published a protocol for doing this type of native fractionation procedure on any lysate. It's a two-part protocol. The first is all experimental, and the second is computational. And so I packaged my code for going from elution profile to set of interactions and set of complexes into this next flow pipeline, CFMS flow that can be run on any Linux system. And finally, there's a website, plants.proteincomplexes.org, where you can search interactions or download data. And if you have any questions about any of the interactions or using it, please email me. This was all done at UT Austin and also with some collaborators at Davis and Eunice. This is my main co-author of Iliopoulos, who was the experimental lead on the biochemistry, and Edward Marcotte, who was the PI. And now I will take any questions.
Thank you, Claire. We have a couple of questions. I think you answered one of them at the end of your talk, but it was generally around that the information would be useful for creating complexes in the plant reactome database, and what would be the best way to get this information into plant reactome?
Yeah, correlating, there's some really interesting metabolic complexes that I've been kind of digging into, and it's not like a stable, like these 6 enzymes are always part of the same metabolic complex. It seems to be like shuffling of membership, or maybe like handing off of protein interactions. And I don't fully know how to interpret it yet. I could show that interactions were slightly enriched for sequential like pathway steps, but not that much.
Okay.
I don't, so I'm not exactly sure how to incorporate it with reactome type things.
And then another question is, there are many cross-linkers. did you use them and how did you decide which one to use?
We used the DSSO spec cleavable crosslinker. The big thing was really soaked up by the highly abundant complexes. So we're really seeing like CCT and photosystem 2, but the coverage of lower abundance things was just not there for us.
And then is there similar co-fractionation available for organelle proteomes?
Not from my PhD lab, but maybe other labs. Yeah.
Okay. Fantastic. And I think with that, we can transition to our last speaker, and thank you for your wonderful talk.